Yes,
Oh,
ma'am, audible.
okay. They were trying to start. Madhu was busy. Okay. It's okay. It is showing live streaming is also available in case if we just cross the numbers. It's only a limited slot that we have taken, sir, because a simultaneous meeting was going on. So it was 100 slot. So in case if,
We are live now and the recording has also been started.
okay.
We can start, I think.
Okay, Anju ma'am.
जी वक्रतुंड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देव सर्व कार्येषु सर्वदा. Good afternoon, one and all present over here. Honorable Speaker of the day, Dr. Beena, respected Principal Sir, Vice Principal Sir, our teacher in charge, Department of Botany, Deshbandhu College, Dr. Aparna Nautiyal, IQAC Coordinator, Dr. Aditya Saxena, Coordinator, DBT Star College Scheme, Dr. Indrakant Singh, Coordinator from Department of Botany, Deshbandhu College, DBT Star College Scheme, Dr. Kumar Shantanu, Convener of this webinar, Dr. Rajni Rajaman Mathur, esteemed faculty members from Department of Botany, Deshbandhu College, and other institutes, and my dear students. On behalf of Department of Botany, Deshbandhu College, I, Dr. Anju Chibber, welcome you all to today's webinar on plant biotechnology, RNAi and CRISPR-Cas9 methods, which are used in targeting specific DNA fragments in crop improvement. Without any further ado, I invite our Vice Principal, Sir, Professor Kamal Kumar Gupta, to address the gathering and motivate our students. Kamal, sir, please.
Kamal sir. Kamal sir.
Do you hear me?
So there is a bit of disturbance, sir, but your voice is coming. It's
Yeah. Do you hear me?
yes, sir. Yes, sir. Yes, sir, we can hear you. Check connectivity issues.
Yeah. Roshni, do you hear me?
Yes, sir, we can hear you.
This unstable
Yeah, I can understand.
So, good afternoon, everyone in India, and good morning in Germany. This is especially to our distinguished guest, today's guest, Dr. Veena. Dr. Veena M. Pavindra, and to hear her. She is Institute for Plant Genetics and Crop Plant Research. Our Agarwal, IQAC Coordinator, Dr. Aditya Kumar Saxena, Indrakant K. Singh, Dr. Roshni Rajamohan, Teacher in Charge, Botany Department, Dr. Aparna Nautiyal, all the of Botany Department for their immense efforts to give the students a platform where they can learn, they can motivate the research, where they have been provided webinars with all the faculty members of the Yeshwantu College and also the students for whom this is specially organized. The talk of the topic is plant technology, RNAi, and Crispr methods. In fact, the RNAi and CRISPR editing, and they have just started, but of course they are going to take even bigger strides in future, where they can really change the entire scenario of genetics and biotechnology. And we have one of the most distinguished scientists in this field. So without taking much of the time, I give the platform to the organizers to continue the event further. Thank you.
Thank you very much, sir. We also have our teacher in charge, Dr. Aparna Nautiyal, with us. She has joined the program in spite of her ill health. So she has also conveyed greetings to you all. She may not be able to speak much. So now I invite convener of this webinar, Dr. Rajni Rajaman Mathur, to please introduce today's speaker. Rajni, ma'am, thank you. Ma'am, you are on mute. Rajni, ma'am,
Okay.
you are on mute.
Okay? Is it okay?
Please unmute yourself. Yeah.
Is it okay? Am I audible?
Yes, ma'am, you're audible.
Okay. A very good afternoon to all present here. A warm welcome to all the chief guests, those who are here on special request they have gathered. A big thanks to Professor Dr. Kamal Gupta, our Vice Principal, and the invited speaker for the day. It has always been a privilege and honor for me to invite, whenever a chance is given by the department, to invite the speaker for the day, especially on behalf of our institution. And this feeling today is really augmented when the speaker happens to be your childhood friend. So I take immense pleasure and pride in inviting the speaker for the day, Dr. Beena Madappattu Parambil Ravindran. We have shared schooling and up to graduation, and then we parted our ways. Dr. Beena is presently working as a scientific associate at Leibniz Institute for Plant Genetics and Crop Plant Research, Gattersleben, Germany. She began her academic career by obtaining her bachelor's, master's, and MPhil degrees in botany from University of Calicut, Kerala, and went on to complete her PhD from Hyderabad Central University. Ms. Ravindran joined as an assistant professor in the Department of Biotechnology at Sir Padampat Singhania University, Rajasthan in 2007, and was an active member in various academic committees of the university. She resigned this well-entrenched job to find contentment in her research activities after receiving the prestigious DART BMBF postdoctoral fellowship sponsored by the German Academic Exchange Service, Federal Ministry of Education and Research, Germany, and further went on to complete her postdoctorate from Institute of Pharmaceutical Biology at Technical University of Braunschweig, Germany. Dr. Beena has been part of many research assignments at various institutes across the world. To name a few: ICAR, Central Tuber Crops Research Institute, Trivandrum, India; Leibniz Institute, DSMZ, Germany; Institute of Pharmaceutical Biology at Technical University of Braunschweig, Swiss Federal Institute of Technology in Zurich, Switzerland, the International Institute of Tropical Agriculture, Nairobi, Kenya, and the International Institute for Tropical Agriculture, Nairobi, Kenya. She is a skilled scientist with so many fields where she has having expertise. Again, to name a few, legal expertise in biosafety rules, techniques in microbiology, CRISPR-Cas9, on which she'll be speaking today, cloning, genetic engineering of plants, tissue culture, greenhouse nurturing of plants, and molecular and in vivo analysis of transgenic plants and evaluation, and many more molecular biological techniques. She has, to her credit, more than 20 research papers, and also is a peer reviewer and member of editorial and scientific advisory boards of many reputed national and international journals. Let me tell you on a personal note, she is also an excellent artist with this pencil and watercolors. So here today, she is amidst us to deliver a lecture on plant biotechnology, RNAi, and CRISPR-Cas9 methods. Now, I would request the audience, before our speaker starts her lecture, any queries that you have may please be posted in the chat box. And after the lecture, the questions will be taken up during the interactive session. So please join your hands and welcome Dr. Beena M. Ravindran. Beena. Dr. Beena.
Hello, everyone. I think I'm audible, or am I muted?
Yes, yes.
Yeah. Okay. I'm really honored to be giving a talk in Deshbandhu College of University of Delhi. And when Dr. Mathur approached me regarding this, I was only happy, and I was like really excited for this opportunity. And I'm thank you very much, Dr. Mathur, for this elaborate, and I'm really touched, the way you explained about me, and the introduction was like, yeah, it brought me a lot of memories, yeah. And I'm very thankful for Dr. Chiba for the welcome speech, and Dr. Gupta, and yeah, thank you for the opportunity. And I don't want to really drag my gratitude and delay it any further. I think I will directly jump into the talk now. So let me share my. Are you able to see my desktop?
Not yet, ma'am.
Okay. Is it not visible, or?
There is a red flowers, I think a small tiny picture we can see. Is that yours? Anthurium, I suppose. It is
One
so
second.
tiny. It is so tiny.
I'm sorry. I gave already that I should share this, but somehow, okay, now you should be able to see.
Yeah. It is the anthurium that you have it. In the picture, the red one. It's so tiny that what we are seeing.
No, it's not the anthurium. One second, I'm trying to.
So it's something. You are maybe you can share your screen.
Yeah, that's what I'm doing.
Yeah.
It's like I'm trying to share my screen. Are you able to see my screen now?
No. It is tiny picture, red flowers. When you are
That's strange. I have already shared my screen.
Madhu ma'am, is there anything at our end?
I don't understand what is the issue with the, I think.
I remember when especially some of the speakers, they find this, their screen comes out very tiny. Many places I've seen this, people having these issues with Zoom. We are all very familiar with this Google Meet. It's a very tiny picture that's coming out.
Ma'am, it's someone else that DP
maybe
or
you
some
can.
picture they have put.
But changing the slides, it's always comfortable doing it yourself.
Yeah, ma'am, now we are able to see your screen.
Yes. Yes. Your entire screen is visible. Vina, it is okay. We can see your screen. If you can hear me. We can't hear you, though we can see your screen. I think her audio. Dr. Beena, can you hear me?
Dr. Bina, your speaker is also showing that it's mute. Your mic is also mute, as we can see. So maybe the audio from the computer is on mute. Ma'am, maybe if you disconnect this your earphone, maybe it will work.
Yes, many a times that also causes. No. Her screen is visible to us.
Ma'am, you can just try to log out and then log in again once more. So maybe it will resolve the issue.
Okay. Am I audible now?
Yes, ma'am.
Okay, then I'm going to talk without my earphones now here. Then let me try to again share my screen. Is it better? Can you hear me now?
Yeah, ma'am, it's better. We can see the screen as well as we can hear you.
Okay, that's wonderful. Oh. I'm very, very, very sorry for this technical problem.
No
Okay,
issues,
now
ma'am.
we
It's not
can,
in our
we
hands.
can,
It's
we
okay,
can
ma'am.
start. Yeah. So I'll be talking today about the technical details of RNA and CRISPR-Cas9. And for the students, I think I'll give a short introduction about biotechnology. I hope you all know that biotechnology is a set of techniques, organisms are used to adapt plants for specific needs. And it can be utilized for sustainable food, new products, nutrition, biotic stress, abiotic stress, protection of the environment, for yield, or even for basic researches in biochemical, genetical, physiological, and other pathways or metabolic pathways, etc. And this can be divided into like 4 different major parts: medical biotechnology, industrial biotechnology, environmental biotechnology, and marine biotechnology. So the major work, what you do in biotechnology is plant transformation, or genetic engineering, or genome editing, as we call it these days. So this plant transformation is can be done using different methods. One is by expression. By expression, you mean expressing a new gene in a plant. By overexpression, you can overexpress an already existing gene, which needs, I mean, like if the product is not as much as what you want, and you want more out of a plant, then you can overexpress a gene and then get maximum out of it. And you can also knock out some genes, which is not necessary. For example, for some genes which are causing some allergic reaction to somebody, you can also knock that out and have a plant which is eatable for everybody. And you can also silence a gene. Silencing is done by just muting one of the genes which you already have. And you can also stack genes, like 2 different genes or the same gene can be stacked. And tDNA integration. By tDNA integration, I mean most of the transformations are done by tDNA integration, but you can also knock out some genes using tDNA integration. So in Arabidopsis, for example, for basic research, you try to integrate tDNA in any possible gene, in between any gene. And if it happens to land in between the exons, then this gene is basically snubbed out. So it cannot express itself. So you can also create mutations like that. And one of the most traditional way of plant transformation is mutation. Mutation can be done by several means, like even by radiations. And mainly it can also be done by radiations, or it can also be done by chemical methods. So when it comes to genetically modified plants or organism, it is usually considered the tDNA integration or Agrobacterium mediated transformation, or by biologist methods, are the genetically modified plant organism. But if you do it by hybridization or by mutation, it is not considered as genetically modified plant. So that is really strange, because in hybrids and mutants, you don't know what you are switching off and switching on. But in all other cases, you know exactly what you're putting inside, but they are considered as transgenic plant or genetically modified plant. But in hybrids and mutants, because it is already happening in the nature, they are not considered as genetically modified organism, even if you do it deliberately. So plant transformation can be done by majorly by 2 methods. One is Agrobacterium mediated and one is biologistics. So on the screen, what you see on the left side is a Helios gene gun, by which you can shoot genes into the plants. So this one is used usually for shooting genes. I will tell you more about this. And this one can be used for particle delivery for in vitro tissues. And Helios gene gun can also be used in vivo, not just in vitro. And here you can see how you produce or how you prepare the bullets for the Helios gene gun. So on the first picture, you will see here that is an emulsion of gold particles in a buffer, which is also coated with the plasmid which you need to send into the cell. And here there is a tubing. You can see there is a tubing, long tubing. So you pour this into this tubing using this syringe here, and then it is equally coated. And this is a cutting machine here. So you can cut it into pieces, and these pieces are the bullets, and these bullets are loaded in a cartridge here. So when the cartridge is loaded, then you can use it for shooting. And once you shoot, you can see that it is empty. So when it is full, you can see here it is filled with gold particles, and here they are totally empty. So that's how you know that your shooting went successful also. So here are the methods how you shoot. You can either put something behind the leaf and then shoot. This is in vivo shooting. And you can also put your hands behind it with a gloves, of course. Even if your hands are bare, it's not going to affect your hands. It is not that that forcefully coming that it can pierce your hands and go inside your tissues. So it's totally safe. And here you can also do it on your desktop or wherever. So it is a very handy gun which you can use. And when it comes to particle delivery system, it has to stand inside a lab. You cannot take it around. So this works with a helium gas filled chamber, which is also stopped by this diaphragm. And there's a this coated, that gold particles coated with the plasmid which you want to put that. Yeah, here you can see the gold particles, and it stands on a diaphragm here. And then when the helium is applied here, it forcefully delivers everything onto the tissue. So you can see that it need a lab, and it need to stand inside a sterile bench so that it can be done. So once it is shot, then the plant can be regenerated by a tissue culture method, and then you can get a new plant or transformed plant out of it. When it comes to agrobacterium mediated transformation, as you all know already, that we utilize agrobacterium, tumefacients or rhizogens for this. Rhizogens is used for creating roots, and tumefacients is used to get plants out of it. Okay, even if you use rooting agrobacterium, you can also get plants out of it, but this is the most common method used with agrobacterium. So this is the basic structure of the agrobacterium we usually use. So there is a TI plasmid which contains a tDNA. This tDNA region is the one which is introduced into the plant cell, which gets integrated there, and then start expressing there, and then it does what we want it to do. So here it gets integrated into the genome. Here only the tDNA region, a particular region which is in between the left and right border. These are the borders. So only these region will get integrated into the system. To have an example here you can see that there is this tissue co-cultivated with agrobacterium. So you can see this white bacterium growing around the tissue. And this is an example of a transformation procedure. So in the first picture, you will see this tissue which need to be transformed, and then the agrobacterium is co-cultivated with the tissue, and the tissue grows. And once it is grown here, you can do the analysis and see. So here, this is a control plant which doesn't have the gene integrated, and this is a positive plant which shows blue color. The blue color, it is showing only blue color because it is meant to show blue color because the gene inserted is a Gus gene. But many people ask that they think they have this wrong notion that every transgenic plant shows blue color, and it is really dangerous to health. And that is also one reason people tend to go against transgenics, but this is not the case all the time. It's just for identification of the plant. So coming to RNAi, a little bit of history about how they found RNAi. So there was an experiment which was done in the University of Arizona by Dr. Richard Jorgensen, and he was trying to introduce very dark purple color into petunia, and he did a transformation with the chalcone synthase gene, and for his surprise, he found plants which are growing with flowers white or a purple plus white. He did not get a very dark purple flower. So that was really surprising. And parallelly, there was another team who were doing also the similar experiment, in which they used one of the genes, DFR gene, to introduce color into petunia. And they also found similar. They also had similar experience that the color did not get augmented, but only got diminished, or they got entirely white flowers. So they found that the flowers are actually having the gene, and it is also getting transcribed. But the only problem is it is not showing up as a product. So they were confused about the results. And parallelly, there was also other experiments happening in rice, this rice. In this picture, in the first row, you can see that when a Gus gene is introduced in its forward direction, it is it is expressing in every tissue here. And when it has a stopper gene or the terminator gene, then also it expressed in most of the cases. And here the first one is a control, and the rest are experiments. And when they put the gene in the reverse direction, then also it expressed. And when they stacked two genes, then also it expressed. But when they put one in forward direction and one in reverse direction, then it was not expressing at all. So there was another explanation given that there is a co-suppression happening. When there are like more genes put inside the plant, they suppress each other and they are not expressing at all. So then in plant community, it was they saw all these experiments, and they were struggling to find a real explanation for this. In animal world also, there were experiments happening, and they were doing it in C. elegans. And here in the first figure, you can see that they put a sense RNA into C. elegans, which is representing a muscle tissue. And they also added antisense RNA into C. elegans. And both the times, they did not find any problem with the C. elegans. They were like giving the same wild type wild type individuals. But when they put double-stranded RNA, that is sense and antisense, together into the C. elegans, they saw that it is producing a twitcher. Twitcher means it cannot it is not having any muscles, so it cannot stay properly, and it starts showing this twitching property there. And they also did the same with the embryos, and they also found that when uninjected, it showed a lot of mRNA inside, and when they inserted the antisense RNA, the mRNA level was a little less, and when they put the double-stranded RNA, it completely vanished. So what conclusions they came into this from this is that the double-stranded RNA can silence genes. So this was possible only by the sense, antisense, and double-stranded experiment what they did in C. elegans. And it silences a gene whose code matches that of the RNA. So in case you're putting a small strand of RNA into the plant or into the animal, and if it matches with another RNA inside the system, then it goes and binds and it creates this double-stranded sense-antisense RNA, double-stranded RNA, and then it gets suppressed. The gene silencing also involves a lot of enzymes in the cells. And for this explanation, Mello and Fire, they got the Nobel Prize in Physiology or Medicine in 2006. And they are the ones who suggested that the double-stranded RNA is the trigger for gene silencing mechanism. So coming a little more closer to the explanation, this goes back to the central dogma of the cell. So here you can see that the DNA is, I mean, there are many theories about the central dogma, but the main one is the DNA transcribes into mRNA, and this mRNA is sent to the cytosol, and then there it is translated and produces the end product, protein. So what happens in RNA interference? In RNA interference, if the DNA gets transcribed, so the DNA is still there, and the mRNA is also there, and the mRNA comes into the cytosol, then what happens there? The RNA, the mRNA in usual case translates into protein, but when there is interference, then the mRNA is getting interfered and no protein is created. So the DNA is there, the mRNA is there, but the protein is not there because the mRNA is getting destroyed. So this system exists in every cell, in plant and in animal. So why is that? The fundamental mechanism of controlling the flow of genetic information is happening through this mechanism, this interference mechanism. So in plants, it participates in defense against pathogens, especially virus infections. And it also keeps the jumping genes in control. And you can see that in every plant or in animals, there are more than 500 genes which are coding for the microRNAs, which are using to block the gene expression. But now we can manipulate this RNA technology into doing what we want the plant and animal to do. So now the RNA technology can be used in research, biotech industry, and medicine, etc. So how is this is actually making? So there is this double-stranded RNA, and it creates a small RNA using a DISA, that is an enzyme, DISA, and then it goes and fixes itself into RISC complex, and I will not go into much into these details, and it will dissolve the sense strand, and only the antisense strand will remain here, and it goes and finds the matching sequence in microRNA, in messenger RNA, and then it is cut into pieces and destroyed. So this is what happens in silencing. So here it looks a little complex, but it is not actually complex when you try to understand what's happening. So when a viral RNA gets into the system, and there is an exogenous RNA, which whatever it is, it can be any pathogen or anything which is put inside the system. So when it gets into the system, then there is also this jumping genes which are getting, which we don't want in the system, also getting its messenger RNA into the system. So what happens immediately? This system gets activated inside the cell, and the microRNA is created, and it goes into the system, and then the RISC complex is activated. This is the main thing, and then it will search for the unwanted mRNA and then chops it down. Yeah. So it is out of its system. So it silences the gene which it doesn't need. So when we engineer it in plants, what we are doing actually is sending tailored dsRNA, that double-stranded RNA, which we want in the plant to be silenced. So this is a tailored RNA which can be sent into the system, and it will go and silence the gene using this CRISP. So that's how we silence a gene inside the system. So I think this is too much of a detail to go into RNAi, so I would just skip this area, and maybe this also. Okay, so there are many small RNAs which are present in the system, and we can use any one of them to create our transgenic plant. So the main ones are like microRNAs, secondary small interfering RNAs, trans-acting small interfering RNAs, NATC RNAs. So all these four can be used, and these are also used. Primary siRNA also is used, and piRNA is PV interacting RNA is in animal system. It's only in mammals and flies. So the microRNA, tRNA, NATC-RNA, HC-RNA, they are all different names of small RNAs, but they all do the same thing. Here, this is the step where everyone matches, and then they go into the mRNA cleavage. So only the origin is different for them. So it is they are called differently only because of their origin. So how RNAi works? It is it may basically gets triggered because of the entry of a long or double-stranded RNA into the system, or it can be because of an introduced transgene or a rogue genetic element or an viral intruder. And the dicer cleaves the double-stranded RNA into short 20 to 25 base pair fragments, and RISC is activated. And once the RISC is activated, it distinguishes between the small RNA strand into sense and antisense. And the sense strand is degraded, and the antisense strand is going and finding the sense sequence. And then the RISC attaches to the mRNA molecule and cleaves it into pieces and silences it. So that's the short story of gene silencing. And the one good thing about RNAi is that the genetic material is intact, and only the mRNA is degraded. And to show an example how we actually utilize it into creating plants which we need, this is an example of tRNA construct. So you don't have to actually know how the construct is done unless you are going to do a research on it. So here you can see that there is a promoter, there's a terminator, and there's a gene which you need to target. Everything is put it in a plasmid, and then send it into the plant. So this is sorry. So by utilizing this technology, you can determine the gene function, you can study the pathways, you can identify targets for RNA screening, and you can also generate knockout models. The main thing here is that it maintains the genome integrity. You are not changing the genetic makeup of the plant or the animal. And it can also provide, when you utilize it in transgenic plants, it can provide broad spectrum resistance against pathogens like viruses and plant stress adaptations, etc. So this is where I would like to stop with the RNAi, and if there is any questions you can ask, or I think I'm in time. So I can take 1 or 2 questions right now, and then maybe we can also have some questions at the end, or I can just continue with CRISPR. How would you prefer?
Madam, you can continue to CRISPR because there is no question, so we can take in the end.
Okay, that would be fine. So I can so erase all your knowledge about RNAi right now, and we can move to CRISPR because they both work in 2 different ways. Okay, CRISPR-Cas9. So what is CRISPR? It is an acronym of clustered, regularly interspaced, short palindromic repeats. So what we see here, these are the repeats. So we have some spacers in between the repeats. So what are they? They are actually the mugshots. They are the mugshots which are taken and placed in between the CRISPR region. So CRISPR region is there in approximately 40% of sequenced bacterial genome, and 90% of those archaea, and it contains at least 1 CRISPR locus. So that means there can be many CRISPR locus in bacteria. So why these mugshots are there, and how they are utilizing it, and why these mugshots are necessary for its defense. So we will see that. So this was a research which was going on since a very long time. So you can see that the genome editing and CRISPR technology is not something which came just now. So it started as far as 1979, this genome editing, and the CRISPR biology started as far as 1987. So many scientists worked on this area to understand why these bacteria are having this CRISPR region, and why there are sequences which are matching other sequences of pathogens or viruses inside the bacteria. So once it was found, it was so amazing that you can see, I will try to explain in simple words. When a bacteria is getting attacked by a bacteriophage or a virus, you need to see this pink genome coming into the bacterial cell. So then the CRISPR region is getting activated. It will take a small piece from this bacteriophage genome and integrate itself into the CRISPR region. So that is a mugshot it is taking from different pathogens. So these mugshots are really used as a mugshot to recognize this virus when it comes again, or if it comes in the visibility, they will immediately know, okay, this is this virus, we have to destroy it. So you can see that these colored pieces here are the viral DNAs collected from different viruses. And in between, what you see are the repeats, which are the CRISPR repeats. So when it is expressing, the mRNA is created and then chopped into pieces with 1 piece of CRISPR region and 1 piece of the viral region. So this is called the CRISPR RNA. So the viral region and the CRISPR piece is called the CRISPR RNA or cRNA. And there is a trans-acting gene which is immediately recognizing the activity in the CRISPR area, and it will start initiating its own expression and will create this trans-acting RNA or tRNA. And this tRNA comes and joins this CRISPR RNA, and when they are combined, they create something called tracer RNA, or here we are talking about the pink virus now. So It's called the tracer RNA. So once a tracer RNA is created, once these three components come together, immediately the Cas9, that is the enzyme which is involved in CRISPR system, it comes and binds to it. And once it gets binded into the Cas9 enzyme, they will start looking for its enemy. So they will start scanning the entire genome for the enemies. Once they find the viral genome there, it will attach the place where it is matching the sequence, and it will unwind its genome or the chromosome, and there it will attach. It will attach and then make a double nick. So usually enzymes do only single nick, but this Cas9 can do double nick. That means both the strands are getting cut, creating small little pieces of the viral genome, and it gets destroyed inside the bacteria. So then it gets immunity against this particular virus or the pathogen. So this is how CRISPR-Cas system works in bacteria. So now we see this and we immediately wanted to utilize it for our benefit in crops and human medicine. Yeah. So to summarize this CRISPR, how it works, the first stage is adaptation, and the second stage is expression, and the third stage is interference and killing the pathogen or the gene which we need to express. So the components, when we want to talk about the components of the CRISPR, it's very simple. It is the CRRNA, the tRNA, and combined together the tracer RNA and the Cas9, and together the spacer and CRRNA and tracer RNA together can be called as a single guide RNA. And we also have this Cas9 enzyme and a PUM. So this PUM is very important. We will come to PUM later. And there are also Cas9. I'm always talking about Cas9, but it is not just Cas9, which is the enzyme. So this is what I'm talking about. But this is a type 2 class, type 2 enzyme, which we usually use in our transformation experiments. But you can also have a lot of Cas genes or Cas enzymes like CPF1 or CMR. But mainly it is Cas9 which is used. Only because it is very easy to use. So coming to PUM, what is PUM doing here? And what is actually PUM? PUM is a protospacer which has homology to the pathogen or the virus. So I talked earlier about sorry. About the CRISPR region taking a small piece of virus into its system. So it is not cutting any piece from the virus, it is cutting a piece which is next to a protospacer. That is a PUM area, protospacer adjacent motif. It is this motif is only 3 codons, like TGG or NA or CNN or something like that. So it could be anything. So this is the so here you can see that this is the protospacer area. That is the small bit of piece which the bacteria takes into its system. It always takes the piece where there is a PUM. So when it want to cut the virus into pieces later, it need to recognize the PUM region first before it can attach itself to the protospacer or the small bit of piece which it has taken. So that is the utilization of a PUM region here. So without PUM, it cannot recognize the virus. So only by seeing the PUM region, the protospacer adjacent motif, it can recognize the virus. So there are many motifs for different Cas enzymes. So it can be different. And as I said, we use usually Cas9. And when we use Cas9, it is always NGG. So it could be anything plus GG. It could be, this N can be replaced with the AT or GC, but there should be two G's next to it. So this is the motive we have to take care when we are creating a construct to do a transformation using CRISPR-Cas9. So the development, how they developed it into genome editing. So what was the idea behind it? So we have this spacer region, as I said, as 20 nucleotide region. And then we have this CRRNA, that is a CRISPR RNA region, small piece. And then when we have this naturally occurring tRNA, which when combines with the CRRNA becomes a tracer RNA. And then what we are doing in our transformation is like removing the spacer region here. We remove the spacer region there and then put our own nucleotide there. The nucleotide should be like 20 to 23. Basically, it is 20 nucleotide. So we put our target instead and then send this into the system or plant or animal, whatever. And then it will cut, it will make the double nick and make a mutation there, creating a silencing effect to the particular gene which we are targeting. So for explaining this method for the first time and also showing how we can utilize it in our experiments, the 2020 Nobel Prize in Chemistry was given to Professor Charpentier and Professor Dorna. So what they did is like they added a small little piece here, this one. So it became very easy to clone into plasmids, so that we don't need much of a material to make a CRISPR gene transformation. You only need this area here. Together, it is called a single guide RNA and a spacer. So then you can turn off any gene, or you can mutate, or you can edit a gene according to your wish. So this adding a small little thing on the top, this 4 nucleotides, GAAA, helped a lot in creating the CRISPR transformations. So how they actually did that, as I said before, there is this Cas9 enzyme which is attaching to the target, and it is unwinding the target area and making double nick. And this double nick gives to the mutation we need. And what they actually did was like adding a linker loop here, and then it became very easy. So this is again a little more expanded explanation for the same stuff. And how much specificity this has when we send this small little piece into a system. Why is it not cutting everywhere, or why is it cutting only this specific spot? Or can we have in a genome that many places has a similar, because we are using just 20 nucleotides, so this 20 nucleotide can be anywhere in the genome else present, and it can cut that also. So it can have off-target problems. But this is not happening. Why is this not happening? It is because of the PUM region and the specificity of this 20 nucleotides what we have. So if there is no PUM region, it will be immediately kicked off from that genome area. And if there is only PUM, it will stay for a while, and then it will be kicked off again. So it goes on and goes on. So if you have this PUM area plus the spacer region, which is completely matching the target, only then it will not be kicked out, and it will cut the genome and making a mutation there. So as the target specificity increases, it will cut the region. Otherwise, it will not cut the region. That's how it gets its specificity from. So what happens when both of the strands are cut? So usually when one side of the strand is cut, the other strand is there as a template for the repair mechanism to work on it. And if there is an A, it will add a T, and if there is a G, it will add a C. So you will know what to add there. But when both the strands are cut, the system will not know what to add there. So it has to repair because it need to be repaired. So what it does is, like, it will add anything, but it feels like adding. So it might add or delete some nucleotides before repairing it. So that's how we get the mutations. So sometimes it is just a reading shift. Sometimes it is a complete failure of production of the protein. So in whatever happens, it is then silenced. So here you can see that this is a non-homologous end-joining repair mechanism. So it either removes or introduces some nucleotides, creating a mutation here. So it can also be done in different way by giving a donor DNA. So if we ask this particular system to replace this area with another gene or another portion, so it will do that. This CRISPR-Cas system can do that. And it will create a new DNA with whatever we want to introduce there. So this is homologous DNA repair, and the other one is non-homologous end joining repair. So in the new DNA, in this method, we can also repair if there is some mutation which is not needed, or for example, in medical industry, they could repair the gene which creates the blood, but a sickle cell anemia, and they created a man. I mean, they did not create a man. The man was suffering from this disease and sickle cell anemia, and he could be cured by correcting this DNA. So such things can be done using this homologous directed repair mechanism. And if you just want to switch off a gene, you can also use non-homologous end joining method. So this is how we utilize the system for our own benefits. So CRISPR-Cas9 applications are wide. Actually, it can be used for DNA detection, DNA insertion, replacement, modification, labeling, and for RNA targeting, etc. And CRISPR-Cas9 applications can be in biological research or research and development or human medicine, agriculture, etc., etc. So we are yet to find out where all we can utilize this still. So this is an example of a pipeline of an experiment, how it happens in plant. So the first of all, what we have to do is to design this sgRNA, that is a single guide RNA. And then you need to clone it into a vector, that is a plasmid. Sorry, I'm trying to take my pen. Okay, so you can insert this single guide RNA, what you created, into a vector. This vector can be any vector, usually used for normal Agrobacterium transformation. So what you need only is this sgRNA in the system, and also the Cas9 gene next to it, nothing else. So then you can do the transformation. And in this case, what as an example, what I'm showing you here is Arabidopsis, which is transformed using RFP gene, that is a red fluorescent gene, just to show how it is done. So in the T1 plants, you will select the glowing seeds, and then grow into the next generation. And in the next generation, if you choose the non-glowing one, you will find the mutated plant, but without the construct itself. So you can also there is a long-standing concern that when you want to use a transgenic plant for eating, then there is also a lot of genes which are coming along with that, like the selection marker genes or whatever gene we put it inside, along with the particular gene which we want in the crop. So in this system, you can also avoid whatever comes along with it and only utilize the mutation. So when you have this kind of selection system, then you don't have the CRISPR, this area in your plant. You will get a pure plant with the mutation. So now the CRISPR-Cas is like it just came five or 6 years back into the system, and we already have a lot of adoptions happening. So from 2012 to 2016, it has been adopted in various varieties of animals and plants. So the current achievements in crop genes, crop system is enormous. You can see that from this pie chart here. And another very good example from one of my friends is the banana which comes with the brown streak. They also used it to destroy the virus behind the banana genome, and you can see how clean this banana is without any streak on it. So these are the benefits in agriculture, and for casual readings you can use one of these papers. It is very interesting. Yeah, that's all about CRISPR-Cas also. So now we can open the question session, and it doesn't have to be completely pertaining to scientific questions. You can also ask anything outside the scientific area. So any kind of questions are welcome. I hope I will be able to answer them, but yeah, questions are welcome.
Thank you, Dr. Bina, for wonderful and knowledgeable talk. Madam, on behalf of audience, I would like to take few questions.
Yeah, please.
What is the difference between miRNA and siRNA?
Yeah, microRNA and siRNA, basically there is no difference. They are called differently. MicroRNA can be, as I said, it is only the origin.
Depending on the origin, yes, ma'am.
Yeah, depending on the origin, where it comes from.
What causes co-suppression of a Gus gene?
Suppression of a Gus gene?
Yes, I think some student has written this.
Okay.
Gus gene, actually it is a kind of marker gene, and
yes,
yeah, we used to transfer for selecting a transformants.
Mm-hmm. Yeah, I know that.
Yeah.
But
for students, I'm telling you.
Yeah, but
Yeah.
Suppression of Gus gene, actually suppression of Gus genes do not happen. If it want to express, it expresses. If it is integrated properly, it is going to express itself. But if you want to suppress it, you have to use other methods to suppress it. Like you have to create another transformant which can suppress the Gus genes. Actually, we were doing some experiments on this by mutation breeding. But in that case, also it takes many years to to really find a suppressed Gus gene. So I don't know if I got the question properly, but yeah, if it is expressing, it is expressing. There is no.
Yes, it expresses. Yes, ma'am.
Yeah.
So we can go for next question.
Yeah, sure.
Does RNA interference occurs naturally?
Yes. That's why we are utilizing the system.
Yes.
For our transgenic needs. Yeah. It is a natural system.
Yes, ma'am. Next question is, what is the difference between restriction enzyme and CRISPR-Cas9 enzyme?
Yeah, a CRISPR-Cas9 enzyme works only when the CRISPR is activated. And CRISPR-Cas9 can make the double-stranded nick into it. And the restriction enzyme works entirely on a different platform. So restriction enzyme can also be used for genome editing, that's for sure. It can also cut apart this genome into pieces, and then you can put the sticky and pieces into it, and you can clearly create another DNA altogether. That's how we do clonings, right? So restriction enzyme can also be used for the same purpose, but with Cas9 you have the ability to utilize it in vivo or in vitro in plants and animals. So that's a difference.
Thank you, madam. I hope it's clear to audience. Is
Yeah,
RNAi applicable to control COVID?
RNAi
Can we use RNAi? I don't think. What do you say, ma'am?
It's too early to talk about that. I think. Yeah, we might find a way to suppress, but yeah, it's too early.
What is the success rate of CRISPR-Cas9 technology?
It's I would place it in between 90 and 95.
Thank you, madam.
Percentage, yeah.
I think I have done with all the questions. And for the students, I would like to ask 2 questions, madam.
Yeah, sure.
With RNAi technology, how much of gene expression can be silenced, and how different transformants vary in gene expression?
Sorry, could you please repeat the question?
With RNAi technology, how much of gene expression can be silenced? It's like 50%, 80%, or 90%, up to how much we can achieve?
100%. 100%. You can achieve 100% silencing.
Okay, next question, ma'am. Can we use CRISPR-Cas9 technology in a plant where genome is not sequenced completely?
It's a risk because you will not be able to screen the whole genome to see if there will be an off target. So that is the only only problem when it is not completely sequenced. But you can definitely use it, and if you are sure that this particular gene is not repeated anywhere, or if it is not a tandem gene or something like that, then you can totally use it. But there is always a risk that it
but
can.
It's always risky to have off targets.
Yeah, you can have off targets if the sequence is not completely known.
Yes, ma'am. Ma'am, thank you very much for answering all the queries so patiently. I'm sure that we all have enjoyed the lecture and expanded our knowledge about CRISPR-Cas9 technology. Now, I would like to hand over the session to Dr. Mathur for vote of thanks. Over to you, ma'am.
Thank you so much, Dr. Abhay. And before starting my concluding remarks, I would like to request humbly to all the participants that please, the feedback link will be provided shortly in the chat box. So you are supposed to fill the feedback, and after that, you will be receiving the e-certificate of participation in your inbox. In case if you are not able to receive the certificate within a few hours, you can reach us so that we can look after that you get your certificates. So now it's time for some closing remarks. Above all, on behalf of the organizing team and participant audience, I would like to express our gratitude and appreciation to our distinguished speaker today, Dr. Beena M. Ravindran, for a well-informed talk. This part has been studied by the students in our paper, Plant Biotechnology, but yes, they have just got the essence. They did not understand the topic well, but you have given such elaborative and very informative talk today. So I am definitely, I now know that they are well versed with this topic. And thank you for clearing all the concepts about RNA interference and CRISPR-Cas methods, enhancing our understanding about the topic. Thank you, ma'am, for sparing the time. Even you are on the vacation, but you have given us your valuable time. We are again thankful to you. We also extend our gratefulness to our patron, our respected principal, Professor Rajiv Agrawal, sir, for his generous support all the time. Due to some prior commitments, he is not able to join today, but he has expressed his wishes for the participant as well as for you, ma'am. We express our humble appreciation to our respected Vice Principal, Professor Kamal Kumar Gupta, sir. Thank you, sir, for your motivation, your encouragement, and your support all the time. We also express our gratefulness to IQAC coordinator, Dr. Aditya Saxena, sir. He's a passionate and untiring efforts of his. They always motivate us to do better and better always. Our sincere gratitude also goes to Dr. Indrakant K. Singh, coordinator of DBT Star College scheme. Any event cannot shape without a hardworking leader. So we extend our heartfelt gratitude to our teacher in charge, Dr. Aparna Notyal. She was not well, but yes, her wishes are always with us in the form of support and encouragement. A dedicated team is the backbone of every event. So we express our gratitude to the team Botany, all the faculty members from Botany and all the students from the Botany Department. Above and beyond, we are highly thankful to our student and faculty participants from different college, from different departments and institutes. And thank you so much for attending the webinar with such a zeal. We also convey our heartfelt gratitude to the technical team at Deshbandhu College for smooth execution of this event. We express our gratitude to our non-teaching staff as well. They help us in many ways. And I sincerely apologize if I have missed out on someone. Thank you again, everyone. In the meantime, the chat box, I think the feedback form is shared, or let me check if it is not. I will request Anju ma'am to kindly share the feedback link. And I request all the participants to switch on their cameras, and so that we can take some screenshots and make a memory to cherish.
Madhu, I think we had Professor Geeta Saxena also, and she had, in fact, had a query if we are having some time. Like,
okay, ma'am, I will see that she can unmute the mic. मैम का क्या गीता's iPhone से है? मैम connected है?
I think so.
Hello, yes.
Hello, ma'am.
Hello, yes, I'm connected with iPhone. So I just wanted to say hello to Dr. Beena, because it's very nice. She's continuing her life in Germany. Even I was a DAD stipendiatin at Freie University at Berlin. So I'm so happy a DAD fellow is flourishing so much, staying in Germany, doing great work. So I'm so overwhelmed. I'm very happy that I'm somehow, you know, your RNA and CRISPR has motivated me to join this webinar. And it was wonderful, and you're very, you made all the concepts so clear. But I had a question like, have you started using this technique in your institute, and on which crops your institute works?
Yeah, actually, I was continuously working on cassava for more than 10 years, and I was using RNA there. And then I used CRISPR-Cas system to knock out a 10 kb gene from Arabidopsis, because it was very difficult to do transformation and knock out these 3 genes, because they were tandem repeats. So this we achieved. And now I am doing CRISPR-Cas in punicum, that is a pomegranate, and also in a medicinal plant which is called hypericum. So these 2 are the ones which I'm working right now. So
great,
we are using CRISPR, yes.
Yeah. So you have become full-fledged German.
Maybe 75%, yes.
Yeah. Okay, that's good. I'm happy. So alles Gute und vielen Dank. Sehr schön. Ja, Sie haben sehr schön gesprochen. So wunderbar.
Ja, vielen Dank auch. Ja, das war ganz gut.
Thank you so much, although we did not understand the last
conversation. Yes.
Yes. So the feedback link has been floated, and we will give 2 minutes, and then I will request the host to, okay, I'm the host, I will end the meeting. In the meantime, if anyone...
The photograph is done, Madhu?
Yes, ma'am. We have taken the screenshots.
Okay. Thank you.
So if anyone want to express, in the meantime, for 2 minutes, we are still on.
Yeah, for the students, I would like to, I would like them if they can contact me directly if they have any doubts or anything, or if they want to know about studying in Germany or working in Germany or whatever. It can be anything. If they want to contact, I'm free. So I'm free for them. So yeah, they can contact me.
Wonderful opportunity, students. Please note down. We will share the mail ID, ma'am's mail ID, so you can always contact her. Thank you so much, ma'am, for this offer. Thank you.
Yeah. Also for the professors, if they are interested in some collaborative work or whatever, yeah, we are open. So.
Everyone, again, for participating in this talk, and thank you so much, Beena ma'am, for giving your time here. And it was a pleasure to listen to you and to know you. Even you are sitting in the caravan and giving us a very wonderful lecture. So I hope the feedback form has been filled by everyone by now. So we wish joy and happiness and health to everyone. And with these lines, we would like to end the meeting. Thank you so much. Namaste.
Thank you so much, Beena. On behalf of Dr. Aparna also, who is unable to be with us because of health. Thank you so much on behalf of the college, entire college. Thank you.
Yeah, thank you too.
Thank you, Madhu, and all the participants. Yes, we can